Glutamic. So there is a requirement of 5 micrograms per liter of biotin in the medium. We have discussed about the importance of addition of urea and the optimum temperature. This is the course of production of glutamic acid. That means whatever the events that happen, whatever the changes that happen during the incubation period. So glutamic acid, it is a primary metabolite. It accumulates in the medium approximately after 10 hours following the inoculation with cells of Corynebacterium glutamicum. There is rapid utilization of glucose with the synthesis of glutamic acid, and that continues for 24 hours or unless for next few hours unless there is depletion of glucose. So we can see here this is a concentration of sugar which is getting depleted, which is getting decreased, and there is synthesis of glutamic acid following. This is the logarithmic growth phase. So these are synthesized during active growth phase. Then the important thing is that there is intermittent supply of urea. That means there has to be regular addition of urea. So it has to be supplied intermittently to facilitate the glutamic acid synthesis reaction, that is reductive amination, that we'll see in the next slide. So fermentation may last for 24 to 48 hours, that is for 2 days, unless there is consumption of at least 40% of sugar to glutamic acid. So this diagram shows the course of production of glutamic acid. To understand the biochemistry behind the synthesis of glutamic acid and to understand the importance of addition of urea at regular intervals in the production medium, we need to understand the biochemistry. So we know that this glucose is hydrolyzed through glycolysis pathway, and it gets finally, it leads to the formation of the intermediates that will enter into triose phosphate. It will enter into pentose phosphate. So all these pathways that breaks down the glucose, it leads to formation of acetyl CoA. Then this acetyl CoA, it will enter into the next cycle. This is called as TCA cycle. So there is formation of citric acid, isocitrate, succinate, fumarate, malate, oxaloacetate, and citrate. This is citric acid cycle. Now one of the intermediates of citric acid cycle is the cycle is citrate, isocitrate, alpha ketoglutarate. Alpha ketoglutarate is then converted to succinate. Succinate is converted to fumarate, malate, oxalate. This is TCA cycle, including alpha ketoglutarate. Now, in this pathway, it is shown separately, but in TCA cycle, this alpha ketoglutaric acid is one of the intermediates of TCA cycle. Now, what happens? This organism whose biochemical pathway is shown in this diagram, that is Corynebacterium glutamicum. One property of Corynebacterium glutamicum is that it requires biotin for growth, and if it is grown in biotin limiting concentration, it will excrete glutamic acid. Now why it will excrete glutamic acid? Another property of this organism is that this organism lacks the enzyme of TCA cycle, which is required for conversion of alpha ketoglutarate acid to succinate. That is, alpha ketoglutarate dehydrogenase enzyme. So that enzyme is absent in Corynebacterium glutamicum. That is another property of this organism. So as this enzyme is absent, there will be no conversion of alpha ketoglutarate acid to succinate. Instead, alpha ketoglutarate acid is converted to glutamic acid through the action of glutamate dehydrogenase. So this is the pathway for the synthesis of glutamate. It is synthesized from alpha ketoglutarate. Alpha ketoglutarate is the intermediate, or it is formed from isocitrate, which is the intermediate of this pathway. Now you see, this is not TCA cycle. This is something else. This is glyoxylate pathway. So if TCA cycle is not working, the another pathway that works is glyoxylate pathway. So what happens in glyoxylate pathway? Isocitrate is converted to glyoxylate and succinate. Then this succinate will get converted to fumarate, malate, and oxaloacetate will be regenerated, and the cycle continues. Glyoxylate gets converted to malate with acetyl CoA. So when it combines with acetyl CoA, it generates malate. This is how the intermediates which are required for the synthesis of isocitrate are regenerated through glyoxylate pathway. This is how whatever the alpha ketoglutarate is generated, it is solely converted to glutamate, and therefore that is the reason why this organism produces glutamic acid in excess, because there is no conversion of alpha ketoglutarate to succinate. That generally happens during TCA cycle in presence of alpha ketoglutarate dehydrogenase enzyme. So that is the benefit of absence of alpha ketoglutarate dehydrogenase enzyme. The organism is getting that alpha ketoglutarate is getting converted to glutamic acid through glutamate dehydrogenase. Now this is the biochemical reaction. This is alpha ketoglutaric acid. You can see this is dicarboxylic acid. That means it has two carboxyl groups, COH, CH2, CH2, CO, COH, alpha ketoglutaric acid. This is converted to glutamic acid. Now this reaction is called as reductive amination reaction. There is reduction, that means there is addition of hydrogen atoms, and that's why it is called as reductive. And there is addition of amino groups, that is why it is called as amination, reductive amination. So we can see here that NADPH, it donates hydrogen atom here, and it gets converted to NADP, and there is a removal of one molecule of water. NH4 is added here, that is NH2, and this becomes glutamic acid. You can see the basic structure of amino acid. This is central carbon, hydrogen group, amino group, carboxyl group, and this is R group. This is how, so this explains why there is a huge requirement of urea. So urea is supplied to serve as a source of NH4 ions, which are required for the synthesis of glutamic acid from alpha ketoglutarate through the action of glutamate dehydrogenase enzyme.